Last Monday of the term? Hey, that's right. So, today I want to finish up one more topic about gene expression, and then we're going to turn our attention to nitrogen metabolism. And as I've mentioned before, it's not going to be emphasis so much on the metabolism, although there will be some pathways I'm going to show you, as much as it is the nitrogen movement. And we'll see a little bit about why that's an important consideration. But first I want to finish up talking about something about gene expression that I've alluded to before, but that is not immediately apparent about how or why this is a mechanism of gene expression. The mechanism I'm going to talk about is called RNA interference, and it's something that we've learned a lot about in the past 15 years. RNA interference is a phenomenon that we see in higher organisms. It was discovered first in plants, but it's also present in animals such as ourselves as well. It probably evolved originally as a protection mechanism for cells. So RNA interference is a way for a cell to protect itself against certain types of viruses. That's probably the first function that it had. But later, cells used the mechanism involved in RNA interference for other purposes, as we will see, and some of those help to regulate gene expression. So that's a consideration here. It's going to be, I think, first apparent that the protection against viruses is something that's fairly easy to understand. This figure I happen to like a lot. It shows both processes on it. That is, the protection against viruses on the left and the control of gene expression on the right. So let's focus for the moment on the left at the protection against viruses. Viruses come in a variety of forms and have a variety of genomes. We haven't talked about them a lot, but some viruses are DNA viruses, meaning that their genome is of DNA. That DNA in some cases is single-stranded. That DNA in other cases is double-stranded. I did my PhD work on a double-stranded DNA virus known as adenovirus that causes flu and cold-like symptoms. Other viruses, however, like flu and measles, are RNA viruses, having genomes that are comprised of RNA. And like DNA viruses, RNA viruses can be single-stranded or double-stranded. If they are single-stranded, even single-stranded viruses will have a double-stranded phase that they go through. So double-stranded RNA is a wake-up call to a cell that there's something amiss. There's something amiss, all right? And most likely, although as we will see there are exceptions to that, but most likely there's an RNA, there's a viral infection going on. So having a mechanism to protect against that infection is very, very beneficial for cells evolutionarily. Well, we can see that occurring here on the left. Here's a double-stranded RNA that's in the cell that's come from a double-stranded RNA virus or a single-stranded RNA virus that became double-stranded. The cell has a protein called dicer that can take that double-stranded RNA and chop it up into little pieces. And the exact size of those pieces for our purpose doesn't really matter, but I'll tell you they're on the order of about 20 to 25 base pairs in length. Okay? Those little pieces are cut up by dicer, and one of those strands is peeled apart to make a single-stranded RNA, as you can see right there. And that single-stranded RNA is paired or sort of absorbed by this protein complex called RISC, R-I-S-C. Okay? We'll see on the next slide that RISC is very important in the being able to control or to destroy the target virus. Okay? But suffice it to say that right now the first part of the protection mechanism is chopping the double-stranded RNA into pieces and then taking one of the strands of those pieces and pairing it with RISC, and that RISC will play a role in the next process. Well, cells saw that they had this mechanism. This is my conjecture, I don't know this, but cells see that they have this mechanism going along here, and they see that double-stranded RNA could be used for some purpose for regulating how much RNA is in a cell. This is a way of knocking out a virus might a cell be able to use double-stranded RNA as a means of controlling how much of a given RNA it has. Remember that with control of gene expression, what cells are trying to do is produce the right amount of messenger RNA as needed for making the right amount of protein. So if a cell has too much RNA, it might be nice for the cell to have a way to reduce that amount of RNA very specifically, and it turns out that this mechanism provides that. So over evolutionary time, cells evolved sequences within them that, when synthesized, could make an RNA that was partly self-complementary. So let's imagine that I've got a gene for, let's say, a DNA polymerase, and I want to be able to regulate how much of that is being made. I could go and transcribe my messenger RNA, and all of a sudden, oh my God, I made too much messenger RNA. If the cell has a DNA sequence that has a portion of that coding region of the messenger RNA that is self-complementary, this could be another location on the genome, it could synthesize this piece, and when it's synthesized, it'll make an RNA that forms a stem loop structure. You've seen a lot about stem loop structures this term. And the stem loop structure might look like this. Well, you notice it's got a little bulge right there, okay? You notice the double-stranded virus over here didn't have any bulge. That's one way of distinguishing the viral infection from a cellular-made RNA. Well, the cellular-made RNA we give a name to, ultimately it has a variety of pre-names, which I'm not going to hold you responsible for, but ultimately it's called miRNA, for microRNA. The precursor to the microRNA has that little bulge, and what that little bulge tells the cellular machinery is that it tells it which strand to use. So you want to peel off this bottom strand because this bottom strand is going to be complementary to the target sequence in a messenger RNA that we're going to basically destroy. So we started from different points. We started with a viral infection on the left. We started with a cellular sequence on the right. But in both cases, we have made a single-stranded RNA that is complementary to something that we want to destroy. And that single-stranded RNA is linked with a complex called RISC. And from this point forward, we're going to see the same thing happen to both of them. Okay? What happens at this point? Well, from this point forward, we have our RISC. That could come from the viral sequence, or that could come from the cellular sequence. And there's that complementary sequence. This, and you say, well, the double-stranded virus is going to have double strands. How does it pair to a single strand? Well, even a double-stranded virus has got to transcribe and make single-stranded messenger RNAs if it's going to make proteins. So we have a messenger RNA that can come either from that double-stranded RNA virus or from the cellular messenger RNAs. It doesn't really matter for this purpose, but we see what happens here is that this complementary sequence can pair, put together with the RISC complex, and the RISC complex has an enzymatic activity associated with it called argonaute. It's a great name, argonaute. Argonaute will cleave the double-stranded RNA, breaking the messenger RNA into two pieces. Well, of course, when you've interrupted the message, you can't translate the thing, and you've knocked it out. If that's a critical viral protein, you've just killed the virus. If that's a messenger RNA that the cell has too much of, it's just reduced the quantity of the messenger RNA. So this mechanism turns out to be very powerful, both for destroying viral RNAs as well as regulating the amount of cellular messenger RNA. Okay, and that's just kind of a summary of all the things that I've told you here. What I would tell you in terms of names that I've told you is distinguish that an siRNA is coming from a virus, an miRNA is coming from a cellular sequence. And we saw that the role that RISC played in carrying that single-stranded RNA which is about 20 or 25 nucleotides long, to a complementary messenger RNA sequence where its argonaute activity breaks that messenger RNA into two pieces and therefore stops the translation of that messenger RNA. As I note here, this phenomenon is widespread in eukaryotic cells. We see it, as I said, it was discovered originally in plants, but we now know it is present in a variety of organisms. One of the things that people are very interested in doing with this technology, and there are actually some drug trials going on, both for humans as well as for plants and other organisms, are using double-stranded RNAs as a means of controlling cellular expression of things. So if we put a double-stranded RNA sequence into a cell that is for a gene, we would like to see what happens if that gene is not being made by the cell, then we could easily do that because that double-stranded RNA stimulates this process I've just described and knocks out the expression of that target gene. And here you're limited only by what you can get into a cell and then see the consequences as a result of that. So there's a lot of excitement right now with using this technology in biotechnology. Okay, and I've shown you this figure before. Now you hopefully have a little better idea about what was happening with that, showing how we're interfering with the translation process. And of course, if you break a messenger RNA into two pieces, no surprise you're going to destroy the translation process. Okay, questions on that before I turn to nitrogen metabolism? You guys have become very quiet in the past couple of weeks. I guess the end of the term spring fever is here, is that right? Yeah. Just what?
It's Dead Week.
It's Dead Week, yeah. Dead Week is the most misnamed week of the term, as I'm sure you very well know. I do try to respect it. Of course, I don't make assignments due during Dead Week. Do cover new material in Dead Week. But I do have some good news, and that is I'm trying to think of something special to do for Friday. So hopefully, and I'll say a little bit about it next time, hopefully I'll have some fun things on Friday for everybody to do. Friday's going to be a little different. Yes. Okay. Let's turn our attention to nitrogen. Nitrogen is one of the more overlooked aspects of biochemistry, in my opinion. Nitrogen is a very critical and limiting resource for organisms. It's also toxic for organisms, so there's a balance that has to happen. Cells want to have enough nitrogen to do the things that they need, but if they have too much in the form of ammonium or ammonia, and by the way, I will use those terms interchangeably. Ammonium meaning NH4, ammonia meaning NH3. How do you make ammonium? You put ammonia in water and you make ammonium. So they're the same thing because we're all aqueous organisms. Ammonium is poisonous. And ammonium is a byproduct of nitrogen movement in the body. So the cell and the body has to be very careful, as I said, to balance how much they've got, how much they need. Well, this isn't about the body, this is about the ecosystem. And the ecosystem is of critical consideration for nitrogen because even though our atmosphere is about 77% nitrogen, that nitrogen is not in a form that we can use to make amino acids and nucleotides, which are the two most common things that we need that contain nitrogen. Okay? In order for us to use that nitrogen that's in the atmosphere, it must ultimately be reduced to ammonium or amine-like substances, and that process occurs in, that occurs in a process like what you see on the screen here. This shows the nitrogen cycle of an ecosystem where we have reduced amines, in this case on the top, coming from the poop, as it were, of organisms that deposit that into the soil. We also see within that soil, the question is, well, how did those amines get there in the first place? Those amines got there in the first place as a result of what we call nitrifying bacteria. So nitrifying bacteria are capable of taking nitrogen from the atmosphere and doing a variety of things with it, ultimately producing ammonium, that can be used to make amino acids and so forth, and we'll see a little bit about how that process can occur. This process right here is why I tell students that we should be very worried about what we put into our water and what we put into our soil. All of the nitrogen that is in you ultimately came through these bacteria. As you pollute and you destroy the ability of nitrifying bacteria in the soil to take nitrogen out of the atmosphere, you ultimately destroy the ability of higher organisms like yourself to exist. Okay? So, taking care and making sure that the soil and these bacteria that live commonly in nodules of plants, for example, that these organisms that are in the soil can live is very critical for life on Earth, because without nitrogen, no amino acids, no nucleotides, etc. So this is one of the reasons people are very concerned about making sure we have a healthy place for these organisms to exist. Balance, as I noted, is critical. Okay? Too much, problematic. Too little, problematic. And we're going to see that nitrogen in the body takes a fairly convoluted path. Okay? A lot of what happens with nitrogen in the body is the movement of amines from one molecule to another through a process I call transamination. That's one mechanism. There are others as well. But transamination is a very critical thing for movement of amines in the body. Amines, of course, being a component of amino acids. The body makes and breaks down amino acids. It makes amino acids, obviously, for the purpose of making protein. It breaks down amino acids for the purpose of energy. Okay? If you, for example, are on a low-carb diet, your body is able to use energy in amino acids to make glucose, which the body must do. But in the process of breaking down those amino acids, the body is producing toxic levels of amines. Nitrogen, as I noted, is needed for the synthesis of nucleotides. All the different nucleotides that we see here is needed for the synthesis of amino acids, and it's needed for the synthesis of non-protein amino acids. We have amino acids in our body that never make it into protein, and these include some of the ones you see here: ornithine, citrulline, and sarcosine. We're going to see both ornithine and citrulline appearing in the urea cycle in just a little bit. And there are other nitrogen-containing compounds that don't appear in proteins either. These include choline, some of the vitamins, and you've already seen carnitine. So, being able to use nitrogen for the purposes of making these molecules is very, very critical for cells. Part of the balance of nitrogen metabolism comes from the ability of the organism to regulate how much it puts out. Okay? So all organisms have a way of getting nitrogen-containing compounds out of the body when the need arises. There are three classes of organisms that we talk about. The ammoniotelic are those organisms like fish that excrete ammonia directly. So anybody who's ever had an aquarium knows that if you don't clean that aquarium periodically, it's going to get pretty bad. The fish will ultimately die because, again, too much ammonia, and that sucker is going to smell pretty nasty as well. And that's because fish excrete ammonia. Uricotelic organisms include birds and Dalmatian dogs, I think I've mentioned this one time previously, that excrete not urea like we do, but instead they excrete uric acid. And you remember that uric acid is a breakdown product of purines. It's also a means of getting rid of excess amines, and it's most commonly found inside of birds. The last group of organisms is the group of organisms that we are part of, and these are the ureatelic, meaning that they excrete urea as their primary means of getting rid of nitrogen. By the way, we also excrete uric acid, so it's not like we don't excrete uric acid, but it's not our primary means of getting rid of ammonia. Most vertebrates are ureatelic, and some invertebrates as well are ureatelic. So you can see the three different substances that are there for excreting nitrogen-containing compounds. You notice that urea is the smallest, and at least the smallest that has two. nitrogens that are there. So urea is a very efficient way for us to excrete urea, and you'll see that as I show you the urea cycle. Before I talk about the urea cycle, I've got a couple other things I need to mention. One is the phenomenon of transamination that I mentioned earlier. So what is transamination? Well, again, as the name tells you, trans meaning movement, amination meaning relating to amines. So we have movement of amines, and this figure illustrates what happens in a transamination reaction. Transamination reactions are catalyzed in general by enzymes known as transaminases, no surprise there, and they catalyze basically the swapping of carbonyl oxygen with amines. So for example, if we look at the top, we start with alpha-ketoglutarate and aspartic acid, and those two swap their respective groups, and when they do that, alpha-ketoglutarate becomes glutamic acid, and aspartic acid becomes oxaloacetate. And of course, this can go upwards as well, so the direction that this goes really depends upon the needs of the individual cell. This movement of amines in this way allows organisms to pass amines from one molecule to another, creating or destroying amino acids in the process. Transamination has nothing to do with the release of ammonia or ammonium ions, okay? Because you can see here we start with an amine, and by the way, an amine is an NH2 attached to something. We start with an amine, or NH3 plus in aqueous solution, and we end up with an amine, all right? So it's attached to something, we don't see it released out into the aqueous environment. That's where ammonia and ammonium ions become problematic. Okay. Now, earlier in the term, I talked about the Cori cycle. And to refresh your memory, the Cori cycle is something that operates between the liver and your muscle cells. When you're exercising and you're running out of oxygen, what happens is your muscle cell converts glucose into lactate, and then it exports that lactate into the bloodstream, where that lactate is taken up by the liver, and through gluconeogenesis is converted back to glucose, which is dumped back into the bloodstream for the muscle cells. And so we have this sort of cycle that occurs. Well, there's a similar and related cycle that occurs in the body. So for example, the Cori cycle, which I described to you, is shown on the right. There's pyruvate going to lactate, lactate going to the bloodstream back up to the liver, where it becomes glucose, and there's the Cori cycle going over and over and over. A related cycle occurs in the body relating to the movement of amines in alanine, and it's called the alanine glucose cycle. The primary difference with this cycle is it involves transamination for movement of the amines, and the intermediate being pyruvate. So if we take pyruvate, for example, and we put an amine on it, as we do on the bottom going to the left, we make alanine. That's the only difference between pyruvate and alanine. All right? Where did the amine come from? Well, some other amino acid. Doesn't really matter at this point for us. But the amine comes from another amino acid, which loses its amine and gets an oxygen by that transamination reaction. Meanwhile, pyruvate gives up its oxygen and becomes alanine by gaining this amine. Well, this alanine carries the amine. This could get now dumped into the bloodstream and go back to the liver. And when it goes back to the liver, the reversal of that process happens. The alanine can release its amine group, become pyruvate, and make glucose. And so basically what's happening in this process on the left is that we're ferrying amine groups to the liver. So the glucose-alanine or the alanine-glucose cycle is ferrying amine groups to the liver. Well, the liver can help, and in fact significantly does help, in the excretion, the release of these compounds into the urine so that they can be excreted. We'll see that the liver plays a role in the urea cycle, which is the way that the body makes urea. And this is a way of getting those amines to the liver. The ammonia part will come later. Okay, so the glucose-alanine cycle gets the amines to the liver. This process here, and this is a little involved, so I'm going to take a few minutes to go through it with you. This process here illustrates what happens by combining transamination, which is what's occurring on the right side of this image, with release of ammonia or the gain of ammonia as shown on the left side. So on the left side we see ammonia metabolism. On the right side we see transamination. Now the title of this slide was the central role of glutamic acid. And that's the number one point I want you to take away from this. Glutamic acid is central to all nitrogen metabolism in our cells, in our body. Okay? It's central to that process. Well, let's look at what can happen, first of all, to glutamic acid. Glutamic acid, if we start in the middle, we can go upwards and create glutamine. The only difference between glutamic acid and glutamine is that glutamine has a second amine on it to make a carboxyamide. It goes on to the carboxyl group on the R group of glutamic acid and makes a carboxyamide, and that creates glutamine. How do we do that to go from glutamic acid up to glutamine? Well, you can see that one way we can do that is by taking ammonium ion, here's this toxic material on the left, putting it onto glutamic acid and making glutamine. That reaction is catalyzed by a very important enzyme known as glutamine synthetase. Glutamine synthetase will mop up that ammonium ion and put it onto glutamic acid to make glutamine. If we want to convert glutamine back to glutamic acid, it's a transamination. Okay? And that's catalyzed by glutamine transaminase. If we have a transamination, remember we have to have an acceptor molecule. Here, this ammonium ion just came from the solution. There was no donor, at least no direct donor, for this enzyme. So we've taken free ammonium ion, and now we've made it an amine, and we're going to transfer that amine to another acceptor molecule. In this case, the acceptor molecule is pyruvate to make alanine. That's like you saw in the glucose-alanine cycle. The very bottom of that cycle showed pyruvate gaining an amine and becoming alanine, and here's how that occurs. Glutamine transaminase. Well, what happens with alanine? It goes to the glucose-alanine cycle, and everything else I described to you happens. So this might be happening out in a muscle cell somewhere. The glucose-alanine cycle gets it back to the liver. Okay. Well, there's other things that can contribute to the glucose-alanine cycle. This is only one. Okay? There's other transamination reactions that can put an amine onto pyruvate and make alanine. It too takes things back to the liver. So we're seeing these things moving back to the liver, and now we get back to the liver and or the kidney, as you can see here, and we see the processes that occur below can actually happen. Here's our alanine coming in from the glucose-alanine cycle. Here is the loss of an amine, okay, through transamination in this case to recreate glutamic acid. That is alanine donating its amine to alpha-ketoglutarate, which is right here. Okay, actually it's not, there it is, alpha-ketoglutarate. All right, donating its amine to alpha-ketoglutarate, that creates glutamic acid. Why do we go back to glutamic acid? Because it's through glutamic acid that we can now split off ammonium. That can be one of the sources of ammonium here. That can be one of the sources of ammonium here. Why do we care about ammonium here? Because we need ammonium to make urea. When we make urea, this can be dumped out through the kidney into the urine and is excreted from the body. Now the beauty of this system is its flexibility. You see, for example, that glutamic acid can go every direction on here. You see that ammonium can be mopped up, in the case on the upper left, as happens in cells. It can be released to make urea in the kidney and liver, as you see down here. Okay? And the products are all useful for cells. Alpha-ketoglutarate, of course, is important for the citric acid cycle. Glutamic acid and glutamine, both important for the synthesis of proteins. And finally, urea, essential for the balance, getting things out of the body if we have too many ammonium ions. So we could expect to see a lot of this process happening in the liver and kidney if the body is in excess amine balance. How might this happen? High protein diet. High protein diet is going to favor this process happening. Nothing wrong with a high protein diet, but it's just going to favor this process happening to get rid of more urea. Okay, I'll stop there and take any questions you might have. Yes? So does this mean a ketogenic diet will have more pressure on your kidneys? The answer is probably yes. Because ketogenic diets will tend to favor more proteins as energy sources, and whenever you have proteins as energy sources, you're definitely going to favor this process. Some people are concerned about the effect of these on kidneys. Okay? I don't know that it's a giant effect, but more protein in the diet as an energy source may in fact have problems associated with amine toxicity. Other questions? All right. Now, I'm not going to ask you to draw this figure, but I think that you should know a fair amount about what's happening in this figure. All right? The central role of glutamic acid. The ability of glutamic acid to both absorb ammonium ions and in the liver and kidney to release ammonium ions. The need to have free ammonium ions to make urea. We're going to see another mechanism of making urea in the urea cycle. But this is a way of mopping up ammonium ions and making urea so that we can get rid of excess balance. The role of the glucose-alanine cycle in carrying things from the tissues, the cells, back to the liver so that this process can happen. As they say, this also happens in the kidney. And then last but not least, all the very common intermediates that one finds in the body. Glutamine, glutamic acid, alpha-ketoglutarate, pyruvate, alanine. All of these things very useful for other purposes than what we see right here. So the glutamate is very central to this overall process occurring. Okay, now I'm going to turn our attention to the least interesting cycle in the body as far as most students are concerned. It's called the urea cycle. And it's a cycle that is like the citric acid cycle. It's the other circular pathway that we see inside of cells. Okay? It's a cycle that occurs partly in the mitochondrion and partly in the cytoplasm, as we shall see. It occurs mostly in the liver, but it can also occur in the kidney. As I said, it's another means of making urea, as we shall see. What else do I want to say? It's not a very complicated pathway, although the structures look complicated, and no, you're not going to memorize the structures, so don't worry about that. I'm only going to hold you responsible for a couple of the enzymes in the cycle. Okay? We see overall what's happening in the cycle, and again, I don't tell you this so that you can memorize it, but let me show you what's happening. We're ultimately taking two amines. We are using carbon dioxide and energy and water to make urea, and then there are the byproducts of all of that. Okay? As we see in this cycle can pick up free ammonium ions. This is not the best figure, but it's the best one that I've got. Let's look at what's happening. Here's the cycle itself. We see it spinning right here. The citric acid cycle spun as well, and if you remember the citric acid cycle, you remember that each turn of the cycle required a molecule of acetyl-CoA. It brought in 2 carbons and had 2 decarboxylations. So we started with a 4-carbon intermediate, we went all the way around, and we ended up with a 4-carbon intermediate. The cycle was dependent upon that feeder reaction, the reaction that put in the acetyl-CoA on each round of the cycle. The urea cycle is also a cyclic pathway, and it too relies on a feeder reaction. And here are some ammonium ions that are coming in. For the moment, we won't be concerned where those came from. It could be the cycle we just talked about. It could be other things that are releasing ammonium ions. Suffice it to say, when there's free ammonium ions, they're going to participate in this cycle. The cell doesn't want to have too much in the way of free ammonium ions around, again, because it's toxic. So the urea cycle is very important in mopping this up. One of the enzymes that is important to understand is the one that catalyzes the first reaction. That's the carbamyl phosphate synthetase, carbamoyl phosphate synthetase, as you can see here. It's basically combining ammonium with bicarbonate. The reactions, the feeder reactions, are occurring in the mitochondrial matrix. Okay? They're occurring in the mitochondrial matrix. A second reaction occurs there in the mitochondrion, and that is the combining of this carbamoyl phosphate with ornithine to make citrulline. Sorry about that. Remember that ornithine and citrulline were 2 amino acids that are not made into proteins. We see them both in the urea cycle. Okay? So the formation of citrulline occurs in the mitochondrion. It occurs by combining ornithine with this carbamoyl phosphate. Knowing the names of those molecules, I think, is important. I'm not going to ask you to know any other molecule out here except for arginine and urea. All right. After this has been made, and you don't need to know the enzyme, after this has been made in the mitochondrion, the citrulline is released, and there's an antiport that puts out citrulline as it puts in ornithine. So there's always a balance. One citrulline out, one ornithine in, and the mitochondrion is happy. Citrulline comes out, and there are a couple of reactions that are made. We're not going to be worried too much about the details of those intermediates that are there. I'll just show you, for example, that we can have a transamination where aspartate is coming in and fumarate is going out 2 steps later. That results in the donation of an amine. Now, let's look at what's happened. We started by incorporating 1 ammonia, and we're incorporating 1 amine. That is an external source, free ammonia, and a transamination reaction that is putting in an amine. The reason I tell you that is the final molecule that's made over here, urea, has 2 nitrogen groups. One of those came from the ammonium, and one of those came from the amine on aspartic acid. All right, so we've got those 2 nitrogen compounds, those 2 nitrogen-containing molecules inside of these bigger molecules. We finally produce the amino acid arginine. Arginine, it can be used, of course, to make proteins. However, if arginine is present in high quantities or the cell needs to run the urea cycle, arginine will be broken down to form ornithine and urea. This is the second enzyme that I think you should know the name of, arginase. Arginase cleaves arginine to make urea and ornithine. Okay? This is our source of urea. Now, one of the things some of you may have noticed when last term you talked about essential amino acids, and it was described that arginine is an amino acid that is in some cases essential, in other cases not essential, and it varies with the age of the organism, and that's partly because of arginine's role in the urea cycle. depending on how much the cell is breaking down arginine to make urea, may affect how much arginine is available for making proteins. And so having an input of arginine from external sources might be important for the synthesis of proteins. Okay, as I noted, everything down here below this green dotted line is occurring in the cytoplasm, and that's where the urea is ultimately released. Questions on that? Everybody stand up. Oh, the question? I'm sorry.
So is there a second ornithine that comes in?
No, that's the ornithine. That ornithine is just being shown transporting across. So it has to get to the antiport, and that's just showing it linked to the antiport. Let's stand up and breathe. Everybody stand up. You look like you're just worn out. It really is dead week by looking at your faces. All right? Stretch. Yeah. I worked in my garden yesterday, and I could definitely use the stretch right now, I'll tell you. This weather is awesome, though, huh? Spring is almost here. Anybody have any good spring break plans? Anybody that your friends all will hate you if you're going to Cabo or something, right? No exciting plans? Just let us breathe, Ahern. Okay. Okay. Okay, let's, instead of going through the individual reactions, which I'm not going to do, and I'm not holding you responsible for, let me just summarize some of the highlights of what I've just told you, okay? The urea cycle is a way of incorporating free ammonium and amine nitrogen to making urea. It's essential for ureotelic organisms to be able to excrete urea, and the urea is made by splitting arginine using the enzyme arginase. Carbamyl phosphate synthetase catalyzes the first of the feeder reactions in the mitochondrion necessary for the synthesis of citrulline. Citrulline is made by combining carbamyl phosphate with ornithine to make citrulline, and all the other reactions occur out in the cytoplasm. Okay? That seems a lot simpler if I say it that way? Okay. All right. There is another way to make citrulline. This is where I'm going to finish today. Okay? Another way to make citrulline. And this one turns out to be important for the phenomenon of signaling. Signaling, okay? Cells, or the body uses a variety of mechanisms of signaling within cells, and one of the mechanisms it uses is formation of this molecule here shown at the bottom called nitric oxide. Nitric oxide is a reactive molecule. It doesn't have a very long half-life, but it serves to provide a signal inside of cells for various things, okay? To produce it, one starts with arginine in a fairly complicated reaction that you can see here. 3 NADPHs, that's a lot of reduction power that's going on. A proton and 4 molecules of oxygen, okay, produces all this mess of stuff down here. And the enzyme that catalyzes this is nitric oxide synthase. The main product of this reaction is this guy right here. A byproduct, as it were, is citrulline, which can be used to perpetuate the urea cycle. Nitric oxide is in a very important signaling molecule, as I said, and it's very important in vasodilation, the relaxing of blood vessels. It affects blood pressure. Okay? It also affects things like the erection of the penis, which requires vasodilation in order to happen. So Viagra works by enhancing the signaling through the nitric oxide pathway, thereby facilitating vasodilation. Okay? So the alternative pathway bypasses those reactions that you saw in the mitochondrion. It bypasses the reactions that you saw in the mitochondrion, going directly from arginine to citrulline. Now, if you think about that, what you've done is you've also bypassed the formation of urea. Okay? So this has some consequences, has some price. Cells don't make a lot of nitric oxide. So it's not like there's a lot of urea that's getting robbed in this process, but there is some that's lost simply because the arginine that would otherwise be used to make urea is being taken away and used to make nitric oxide. Okay. Let's stop there. I said that's where I'd stop. I do have a song for the day relating to the histones that we talked about, the chromatin we talked about last time. What happened there? Minus charges cling to pluses tight. It's a slow one, I'm sorry. Chromatin assembly. I'll let him sing it. Okay, guys, see you on Wednesday.